The title of my dissertation research is Advances in Electrocardiographic Imaging, Technical Improvements and Novel Applications. And this is kind of a big, long, very specific title that describes a very neat, nice little window of what I've been working on. But it also doesn't necessarily give you a clear idea of what I've been thinking about throughout these last few years as I've been doing my research. So as we go through this talk, I think it might be helpful to keep a slightly abbreviated title in mind. And that is Seeing Your Heart in a New Way. Because that's kind of how I've seen this body of research that I've been up to, right? I knew almost nothing about the heart when I came to this lab to begin with, and now I know almost more than I think thought was possible, more every day. And so every day that I do my research, I think about it as seeing the heart in a new way. So with that, let's go ahead and get started, and I can describe you what I've been up to for the last few years. So there's something many people have in common between exercising, taking a test, and public speaking. And it's that makes that thing in your chest start beating harder and faster. Turns out that thing is your heart. And your heart is kind of a lot like an engine, right? An engine helps keep you running, like keeps the car running, gets it where it needs to go. And your heart's a lot like that for your body. It pumps the blood around to keep your tissues supplied with oxygen and nutrients and remove wastes. Now, cars' engines can break down. And when a car engine breaks down, you can't drive the car anymore. Oftentimes, you can fix the engine, keep the car running, but it's way better usually to do maintenance beforehand, to detect the problem before the car completely breaks down. Is there anything that tells us when the car engine's about to break down? Well, oftentimes there's some sort of a system in the car, a check engine light, right, that will come on and tell us we need to have the engine looked at. So what about our heart? Having your heart break down, that's a big deal. You don't ever want to have that happen if you can avoid it. But is there some sort of a similar check engine light for your heart, some sort of check heart light? Well, it turns out that there is, and it's called the electrocardiogram, or ECG. And this is a measurement of the electrical activity of your heart that can be made from the body surface. Similar to a check engine light, the ECG is non-invasive, and it's very fast, and it can give us a broad idea of what's going on with your heart. Now, you've probably seen an ECG recording like this one at some point in the past. Doctor dramas like to put them up in the background. And each of these squiggly lines that you see here is the electrical activity recorded from a different place on your torso. Now, a trained expert can look at these ECG recordings and tell whether or not there's a problem with your heart. So your heart sits approximately in the center of your chest. And to be a little more specific, it looks a little bit more like this with some different chambers and different conduction pathways. And when you go into the clinic to get your ECG recorded, they're going to place these electrodes on your chest in various locations, and it's going to produce a signal something like this, where each of these different waves or these bumps in the signal correspond to different electrical activity throughout your heart. Now, in a doctor's office, they're usually going to use more than one of these. They're going to use 8, 12, some number. But as engineers, we can always think, like, why stop at that many? We can put hundreds of electrodes on the chest and get all the information we want. The problem with this is that it becomes very hard to analyze, right? It's hard enough to look at 8 or 10 signals at once. Now, what are you going to do when you have to look at 100 different signals spread over the heart and try and make some sort of a decision? So the reason I'm introducing this is because it leads us nicely to a technique called mapping that I'm going to use throughout the rest of this presentation in order to interpret these complex bioelectric signals. And what mapping is we take each of these signals and we map it to a color, and then we display that color on the torso. And that looks a little bit something like this, where we have red corresponding to a high value measured on the torso, and blue corresponding to a low value measured on the torso. And these are electrical potentials or voltage. Okay. So using that in mind, we can understand what's going on across the whole chest as a reflection of what the heart is up to. But I just want to remember these signals that we're viewing as this map come from this ECG recording, and that's in terms of time and voltage. And so this map that we're looking at here is a snapshot of what your heart is up to at a singular time instant. If we wanted to, we could take a bunch of these snapshots and string them together into a movie and watch the complex electrical behavior play out across the torso. And it'll look something like this in this video I'm about to show you, where I'm going to show you a representative time signal on the top of the screen. And then I'm going to show you a changing torso potential map. And it's going to play 2 times. The first time a little faster, second time a little slower. Okay, so that complex electrical activity that we can measure pretty easily from your torso is a reflection of what's going on in your heart. And to illustrate that, we're going to look at one more video where we're actually going to make the torso transparent. And we're going to look at what those electrical activity looks like on the heart itself. So this blob in the middle of the chest here, that's a representation of the heart. And what I want you to pay attention to in this next video is how the signals you see on the torso, this pattern of color, is kind of a reflection of what's going on the pattern of color inside on that small surface inside the torso. Okay, so that's great and all, right? But why is it useful? Why did I spend all this time telling you about it? Well, because I want you to understand there's a relationship between the electrical activity that's happening at your heart and the electrical activity at your torso. So that's the key takeaway from that so far. But now getting back to our original problem, where's our check heart light, right? How can we use this information to understand when the heart might be having a problem? Well, a trained physician or any kind of trained expert can look at these signals, such as this body surface map, and they can diagnose things. They can say, I expect the signal to look like this, it looked like that. I think there's this kind of a problem. The problem is that when you just look at the body surface potentials, which you said are very easy to record and very easy to access, it's a limited and indirect signal. Going back to our car analogy, imagine that you needed to diagnose what was wrong with your car by just putting your hand on the hood. Right? Now, there's some expert mechanics out there, like my grandfather, who could stick his hand on the hood of the car and he could tell what's going on with that. But not everyone can do that. And that doesn't work for every problem. In the case of the heart, what we really want is we want to know what the electrical activity is at the heart itself. So this blob over here being a heart surface. And because this bioelectric activity at the heart is the source of what we see at the torso, we often call this the bioelectric source. Now, the pros of this is that the bioelectric source is very detailed. It has the information we need to make really accurate diagnoses, but it's really hard to record, right? We have to stick electrodes into the car, or open up the hood to see what's going on in there. So wouldn't it be great if we could use this easy to access information to get at this more useful information? Back to our car analogy, this is kind of like what you do with those, code readers. You can plug it in and take the easy to access information and it'll tell you something specific about what's happening inside the car without ever having to open up the hood. So is there something like this for our heart? Well, it turns out there is. It turns out using a series of computational modeling techniques, we can understand the relationships between the bioelectric source at the heart and the torso surface recordings. And we can use those to create a system to diagnose using the body surface recordings. This system involves understanding the relationship between how the bioelectric source creates the body surface potentials in what's called the forward problem. And then the more clinically relevant, taking the body surface potentials and understanding how to use those to estimate a bioelectric source in what's called the inverse problem. And all of this relies on some computational modeling techniques that typically involve creating a model of the torso, which includes things like the location of the heart and various other things we'll get into. And this system of leveraging this torso model and these body surface recordings to understand the bioelectric source is called electrocardiographic imaging. Now, in order to understand specifically the inverse problem, which is the clinically relevant one, we need to understand a little bit about the math behind it, or the general formulation. So this math that I just put up on the screen is a general formulation of how we can think about doing the inverse problem. This is just one example of many. And let's go ahead and walk through it real quick. So this first term here, phi h, that's our bioelectric source. That's the thing we're trying to estimate. This next term, A, this is a representation of our torso model. This includes everything like the physics involved with how the bioelectric activity propagates or projects through the torso. This includes the location of the heart, location of the torso, all that good stuff. Phi t, this is our measurement. These are those body surface recordings that we can easily make in the clinic. L is a function that computes what's called the loss. It's like a comparison between two things. So in this case, we're comparing these torso surface measurements and the heart surface measurements times the forward matrix. And what this means is we're essentially saying, I have a guess for what I think that the bioelectric source should be. How close does that guess simulate body surface potentials compared to the ones that I can measure? The next term is term F, is where we impose assumptions about our model. right? This is where we can say things where I think that the bioelectric source should be of a certain amplitude, of a certain spatial or temporal smoothness. And this process is called regularization. And then the last term is lambda. This is a weight. This is how important we say this regularization term is versus our reconstruction term. So this is a way of saying how important I think it is that the whatever bioelectric source I guess matches my assumptions versus matching my measurements. So with all of that in mind, we have a somewhat of a robust description of our ECGI system. And before we get past the introduction, there's, oh, my slides are a little messed up. There's one last thing I want to go over, and that's that this system is what I like to call a rule-based model. And now a rule-based model can be generated by taking things like the physics involved. So this is included in our torso model. The assumptions, so for example, we could assume extracellular voltage is what we're measuring, and extracellular voltage is what we're trying to estimate on the heart. And it takes in rules, such as this equation that we said is how we solve the inverse problem. And a rule-based model will combine all of these things together into one discrete computational model that we can then take to use to take inputs and turn them into outputs. And in this case, using ECGI, we take the inputs of body surface potential maps, and we produce an output of a bioelectric source. Now, ECGI has seen some clinical adoption. There's several companies that have worked on implementing this with varying success, but there's also plenty of technical challenges remaining and plenty of improvements to be made. And with that, let's get into what I chose to do for my PhD, which was tackle some of those technical problems. So the goal of my whole doctoral research was to improve ECGI, and this is split up into a number of aims. The first one is to address common errors in ECGI, specifically errors in the position of the heart used in the torso model. The second one is to address the effects of uncertainty in ECGI by applying state-of-the-art uncertainty quantification techniques. And the last one is to apply ECGI to a specific disease, myocardial ischemia, by leveraging some machine learning techniques. So let's go ahead and get into that first aim. So in this first aim, we specifically focused on correcting for errors in the position of the heart. To understand what I mean by errors in the heart position and why they matter, let's look back at our ECGI diagram. So in this ECGI diagram, we know that we have torso potentials and we want to reconstruct a bioelectric source using the inverse problem. And in order to do so, we have to create a torso model. Between these, this torso model is a computational representation of your body. It includes the position of the heart. Now, this torso model is typically generated to include positions of all the relevant organs and all of the electrodes that we care about. And we usually acquire this using some sort of medical imaging device, such as an MRI, which gives us a 3D position of where all your organs are inside of your chest. However, the heart doesn't stay in one position. And when we take an MRI of someone's body, we only get the position of the heart in that one place that they are. But the heart moves all the time. So when we go back to our ECGI formulation, if from our MRI, we know where the heart is in one position and then it moves, that's going to introduce errors into our ECGI formulation. And you can tell that the heart moves yourself. If you take your hand and put it kind of on your side and lean forward a little bit, you'll feel your heart beating. And it'll be pretty strong, right? But then if you take a deep breath and lean back, you're going to feel that that beating goes down. It's because your heart has moved position. So what you're going to see in this video right here is an example of that. This is a single slice of a medical imaging scan while someone's breathing, and you're going to see the heart beating, but you're also going to see it moving and the shape of the chest changing during that breathing cycle. Of course, it came with sound. So to understand why that motion of the heart matters, let's go ahead and look at the situation where we have our heart inside of the chest, and we have the potentials measured on the heart and the potentials measured on the torso. And we're going to let this heart move. And as it moves, you're going to see that the electrical activity on the heart doesn't change, but the electrical activity that we measure on the torso does change. Oh, someone annotated. Okay. So I'm certainly not the first person to notice this problem, the first person to try and address errors in the heart position. And a lot of the research that we used in this particular aim relies on these previous studies. Specifically, our method for addressing the error in the heart position starts with a number of different heartbeats. Right, so each of these is a recording of the body surface potentials for a different heartbeat, and then an initial estimation of where we think the heart should be. We then take these and use all of these multiple heartbeats worth of body surface recordings to do the inverse problem where we estimate the bioelectric source given all of those. And we do this using a specialized formulation of the inverse problem that we call the joint inverse, wherein we take our usual inverse equation and replace phi t, the measured torso surface potentials, with a bunch of different torso surface potentials. Our second step is to take that estimated body surface potentials and pass it into our position estimation step. And this is where for each of the heartbeats, we try and match the position of the heart to the body surface measurement. So we say, if the heart is here, does that produce a good match with our body surface potentials when we project the heart surface potentials to the body surface? And we do that using a similar kind of optimization equation where instead of guessing what the bioelectric source is, we guess some parameters for the position of the heart. Finally, we iterate this process of estimating the heart surface potentials and then estimating the cardiac position or the heart position until we've reached some sort of criteria that tells us that we're ready to stop. And this is usually that the position estimates aren't changing very much from iteration to iteration. And what we're left with at the very end is a cardiac position or a heart position for every single heartbeat that we put into the algorithm. So in order to test this, we need to have some data. And so we turn to an experimental preparation where we take an isolated heart. It's the heart that's been removed from the body and is supported by an external perfusion system. And we then put it into a torso-shaped tank that's embedded with hundreds of different electrodes. And then we then outfit the heart with some electrodes to record the electrical activity out the heart. So this blue cage that encloses the heart. We then fill the torso with an electrolytic solution that allows us to mimic what a real torso would be like and measure electrical potentials from all of our different recording arrays. Our next step was then to take the position of the heart and synthetically move it to a bunch of different positions to simulate how it might move during breathing or during changing of body position. And to do so, we had to parameterize the heart, which means set up numbers that describe the position of the heart. And we did so using 3 rotations, pitch, yaw, and roll, and 3 translations, X, Y, and Z. And we generated about 100 heart positions in this kind of a range. Now let's go ahead and look at some results for how well we were able to reconstruct these heart positions using our algorithm. So these graphs that I'm about to show you, they're going to have the target parameter, the parameter we wanted to achieve, and the reconstructed parameter, the parameter that our method gave us. On each of these, there's going to be a red line, which represents where the heart started for every heartbeat, and then a black line, which is where the heart should have ended up. This is, if everything was perfect, all of our points should end up on the black line. And then we'll also have a number of different points, one for every heartbeat that we guessed the position for. The distance between the black dots and the red line is how far our method moved the heart for that particular heartbeat. And the distance between the black dots and the black line is how much residual error, how much inaccuracy there was in our method. So looking at that for our first parameter, which is pitch, which is kind of like the motion of the heart forwards and backwards in your chest, we get something like this, where you can see most of the black dots are close to the black line. That means we did a really good job of reconstructing the pitch parameter for all of these different heartbeats. The next parameter is yaw, which is the side-to-side motion of your heart, kind of left and right. It's a rotation. And you see for this one, there's some cases where we did a good job, but especially as the target gets close to 0, it's not perfect, right? We don't do perfect at reconstructing the yaw for every case. The same can be said for roll, which is the rotation of your heart along its long axis. Think about how a rolling pin rolls along its long axis. And we see a similar trend where some of the reconstructions are pretty good, but some of them, especially towards 0, are not as good. The next 3 parameters are the translations. So X is the side-to-side rotation, left to right, or side-to-side translation of the heart in the chest. And we see we do a pretty good job with X. It's pretty close to the line for most of these dots. The next is Y, which is the back and forth translation of the heart in your chest. So from your back to the front of your chest. And in this case, we get the pattern of the parameter pretty good, but we get a little bit of an offset between where it should be and where it ended up. And then the final parameter was Z, which is the up and down motion of your heart. And we see we almost did a perfect job at estimating Z. All of the points are very close to the line. Now, it can be hard looking at these parameters to understand really how good we did, how much lingering geometric error there is, how much error in the position of the heart. And so the next metric we looked at is the localization error. This is the distance between where the heart should be and where it actually is. And we wanted to compare what it was when we started, before we applied our method, and what it was after we applied our method. So before we applied our method, this box plot shows that there was a really wide spread in the localization error, how far away the heart is from where it should be, with an average close to about 40 millimeters. After applying our method, every single heartbeat had an error in the heart position for under 10 millimeters, which is a pretty good improvement. And we'll come back to what the significance of that is later. And remember, the point of the ECGI is to estimate the bioelectric activity of the heart. So our method seems to be okay at localizing the position of the heart, but it means nothing if it doesn't make our ECGI solutions better, right? If it doesn't make our estimations of the heart's activity any better. So the next thing we checked was how well we improved the ECGI solutions. So what you're going to see are some potential maps, again, this time just at one time instant, not a whole movie. So this voltage is showing you what this color map is showing you what voltage corresponds to what color. And we're going to be looking at these reconstructions on the heart surface, but I've taken the heart surface and I've kind of flattened it out so we can look at all of it at once. But remember, this heart surface comes from this 3D geometry that's inside your chest. This represents the electrical activity on your whole heart. And for those of you who are curious, we're looking at a peak at a time point at the peak of the QRS, which is right in the middle of ventricular activation. So I'm going to put the measured potentials. This is our target. This is what everything else should look like if we did a good job up here in the top corner for comparison. And we're going to look at a best and worst case scenario for a couple of different methods. The first off is no optimization. This is where we didn't use our method to estimate the heart position. And we can see in the best case, the pattern matches pretty okay. There's some discrepancies that you can probably see, but the pattern looks okay. In the worst case scenario, though, these two patterns are very different, right? The potential map you see at the bottom and the measured. And so that means we didn't do a good job. This could lead to misdiagnosis or a misinterpretation of the ECGI solution. Next is after applying our optimization method, our estimation of the heart position. In this case, the best case scenario still looks pretty good, pretty close to the measured. It's a little bit different than no optimization, but it is a pretty good estimation of this pattern. How about worst case scenario? Well, the worst case scenario still looks pretty good. It almost looks as good as the best case scenario, right? So that means that we've not only improved the worst case scenario accuracy, but we've also really tightened down on the variability in the solutions. And then last, we wanted to check against the gold standard. What's the best it could have been if we used the proper heart position? And that's what we get in this last column, where when we use the target position, this is what it looks like for the best and the worst case scenarios. And I want you to note that the best and the worst case scenarios for the target position and the optimized look almost the same, right? So we've done about as good as we possibly could. So this has all led to a publication where you can read about all the various other ways that we've tested this method. And with that, I'm going to move on to aim 2, which is addressing the effect of uncertainty in ECGI. So uncertainty quantification is a complex and mathematically intense field of study, but a common way I like to think about it is in terms of baking cookies, something I spent a lot of time doing yesterday. So when you're baking cookies, you have a number of different ingredients like flour, baking soda, butter, sugar. And depending on your recipe, depending on how accurately you measure, depending on how you're feeling that day, you might include more or less of each of these ingredients. There's some amount of error and variability in these inputs. And that can have really tangible effects on your cookies. It can really affect their quality and various other aspects about them. So we kind of want to understand how this input variability can affect our output, these cookies. So if we think about each of these input parameters as like a probability distribution, which is a mathematical way of saying what's the likelihood of a value to happen given, all these different values that could be, then we might get something like this. And then if we consider our output as something like the quality of the cookie, maybe the chewiness, then we could get a distribution that looks like this. It can be really hard to understand how this input variability creates that output variability. And this is where the field of uncertainty quantification and uncertainty quantification techniques come in to help us. So uncertainty quantification is a method of not only generating this relationship between the input probabilities and the output distribution, but also analyzing them to understand things like parameter sensitivity. So which of our inputs is the most important, or which of them has the biggest effect? And then variability of the output, right? How wide is the spread of the quality of our cookies? So returning back to ECGI, let's think about how we might apply uncertainty quantification to this problem we've already discussed. Looking back at our ECGI setup, we know from the first aim that errors can occur in the generation of the torso model, and specifically, those errors can occur when we're localizing the organs. And I'm just going to focus on the heart position. We know that from medical imaging, we get one heart position, but that's not always accurate. And we also know that even after applying our sophisticated methods for estimating the heart position, there's still some amount of error, right? We don't do it perfectly. So the next thing we're led to ask as engineers is, okay, there's some error, but does it matter? Do we care, right? How much does that error matter? And should we, keep seeking zeros in that regard? So others have looked at uncertainty quantification in the past with relationship to ECGI, but most of them have focused on the forward problem, which doesn't give you as direct of an understanding of what the impact of the uncertainty could be in the context of the actual use cases of ECGI. So we wanted to instead look at the inverse problem, which is more technically challenging, but also a more relevant problem clinically. So with that, we parameterize the position of the heart, this time a little bit more simply, just according to X, Y, and Z, which is the side to side, front to back, and up and down motion of the heart. And when we want to plug this into our UQ pipeline, where we have the parameters of the position of the heart as our input parameters, our output or our model is ECGI, and our output is the bioelectric source. And we want to apply uncertainty quantification to understand the sensitivities and standard deviations that result from these variabilities in the position. So before we do any uncertainty quantification, we need to say how uncertain we are in the heart position, right? We want to say, do we think we're within 1 millimeter, within 5, 10, 15? And for the purpose of this study, we said, oh, we want to know what the effects are for all of these different ranges. So we've selected 6 different ranges for uncertainty in the position of the heart, from 1 millimeter up to 25 millimeters for each of these parameters. So we use the same data that we did for AIM-1. So it's the same target potential map, right, at the same time instant. But in addition to a potential map, we also get a standard deviation map, which is a measure of how variable the solution is over space. And it gets a different color map where blue is low variability and yellow is high. And now we're going to look at the inverse solutions and the standard deviation maps for each of these different ranges. And I'm also going to show you what the distribution of each of these parameters looks like on the very bottom. So in this first column, we can see the inverse solution, which this is the average inverse solution given this range of motion of the heart. And we can see the standard deviation map. And from what we can tell here, the standard deviation map is all blue. This means that there's very little variability in the inverse solution when you allow the heart to wiggle plus or minus 1 millimeter. The same is true for plus or minus 2 millimeters, right? Very little standard deviation. And the inverse solution, the average solution changes a little bit. I would argue maybe even gets a little bit more accurate, but doesn't change a lot. We start to see some results at range 3, which is plus or minus 10 millimeters. We start to see just barely a little bit of standard deviation peeking out that we can observe. And remember, plus or minus 10 millimeters, that's how much error we were seeing in our previous method after correcting for the position of the heart. As we continue, we can start to see that that standard deviation increases, as you would expect. If you assume a larger variability in the heart position, then you'd assume it's going to have a more variable solution, all the way up to range 6, where we see some somewhat substantial variability in the heart position. And just to give you an idea of what plus or minus 25 millimeters to 1 millimeter looks like, here's an example where in each of these cases, there's 2 heart shapes that are separated by either 1 millimeter in each direction, 5 millimeters, 10, 15, 20, and 25. Right, so this gives you a sense of how far apart we're talking, how much geometric error we're talking about. The next thing that we get from uncertainty quantification is sensitivity, right? This tells us which of these parameters are contributing to the variability in our inverse solutions. And so just to remind us, X is the side to side, Y is the front to back, and Z is up and down motion of the heart. And we're going to show a sensitivity map from 0 to 1 with this new color bar for each of these different parameters. So we can see for X, this is what that sensitivity map looks like, for Y, and for Z. These patterns can be somewhat complicated to understand, but let's just pick a specific feature that we care about. Let's say that we really care about this region on the measured potential map between this elevation and this depression. That can be useful for calculating what's called activation times. We can see that any variability that's happening in that region is mostly the response to variation in the X parameter. So this could tell us, hey, if the clinician is interested in this particular region, then we need to tell them that the X variability can have this effect on it, or we need to try and control for the X position of the heart. And just as a reference, that area also happens to be one of the areas of higher standard deviation, right? So this particular region, which is affected by X, seems to have some possibly impactful influence on our inverse solution. So we've put together this and a lot of other studies into a paper that was submitted to Computers in Biology and Medicine. And there, once it's published soon, you'll be able to read all about the various other ways we've tested this method, this application of uncertainty quantification to the inverse problem. Okay, now on to the final aim. So a goal of my PhD projects has been to improve ECGI, and I've focused on these three aims mostly with some engineering, a lot of engineering in mind. But let's take a quick look at what the title of my PhD is going to be, hopefully. It'll be biomedical engineering, right? So a lot of the techniques I've focused on so far are very heavy in the math and the technique. Yes, they've had some biology tilt to them because they're applied to the heart. But so far, I've really checked off that engineering box. What can I do to check off the biomedical box? Well, I can focus down on a specific disease. And that's where aim 3 comes in. We wanted to create a data-driven formulation of finite element reconstructed myocardial ischemia. And the words I want to focus on there are myocardial ischemia, right? This is a disease of the heart. Many of you are probably familiar with myocardial ischemia because chest pain is the most common cause of a visit to the emergency room. And chest pain may or may not be related to myocardial ischemia. When you go into the emergency room with chest pain, their first concern is, do you have a heart attack or do you have indigestion, right? And so it's really important for us to not only be able to detect ischemia, but also to classify, where it is, how severe it is, those kinds of things. Ischemia happens when you have reduced blood flow or inadequate blood flow to the heart to supply it with its metabolic demand and to remove waste sufficiently. And it can happen from a wide variety of diseases such as coronary artery disease, coronary spasm, and many others. But is there a check heart light for myocardial ischemia? Well, as you might have guessed, yes, there is. And to understand it, we need to look real quick at some cellular physiology. So the heart, as we see here, is made up of billions of individual cells, these brick-like pink structures you see here, and these cells are electrically active. This is what gives rise to the electrical activity of the heart. And if you were to take a voltmeter, a way of measuring this electrical activity, on an individual cell, you would see something that looks like this, where we see this dynamic changing membrane potential, the voltage across the membrane throughout time. And this is called a cardiac action potential. Now, in the case of myocardial ischemia, the shape of this healthy action potential changes, right? With an ischemic cell looking like the black line, or a healthy cell looking like the black line, and an ischemic cell looking like the dotted blue line. This change, specifically at this time point that I've highlighted here with green, which is called the plateau phase, gives rise to a very characteristic change in the ECG signal that we can use to detect myocardial ischemia. When we have two regions of tissue, one of them, the blue one, which is ischemic, and the healthy one, which is pink, right next to each other, there are currents that happen between the different voltages that give rise to an extracellular potential field, which we can then measure as changes to the ECG, which is either away from 0 in the positive or negative direction, depending on where you're looking. And this is our check heart light, right? We can look at an ECG and we can say, there's ischemia here because we see changes to the ECG. So when you go in and use traditional ECG methods to look for ischemic markers, we can probably see them, right? We can see these ischemic changes, but we don't know always where this ischemia is happening in the heart. We can have a rough guess, but it's kind of hard to tell, right? Remember, you're putting your hand on the hood of the car, you're trying to guess where in the car the problem is. So wouldn't it be great if we could get a more detailed picture of the heart, maybe using ECGI, for example? right? And so if we were to take our generic ECGI model and replace our bioelectric source with transmembrane potentials, which is the source of that change in that ischemic change in voltage that we talked about earlier, and we were to plug in these body surface measurements, the check engine light that we measure from the body surface of ischemia, then maybe we could reconstruct myocardial ischemia using ECGI. To do that, we need to look again at our ECGI inverse equation, and we need to modify it a little bit, where in phi h is now the transmembrane potentials defined across the entire heart, based on whether or not the cell is ischemic or healthy. Now, I'm not the first one to do this. Others have kind of created an ECGI formulation that works on this. And one of the difficulties or technical challenges with this that I sought to address is that you have to define a transmembrane potential everywhere in the heart, right? We want to know where the ischemia is happening throughout the heart. And that's a lot of, oops, sorry. That's weird. Okay, that's a lot of parameters, right? That's thousands of parameters for a given mesh of the heart or a computational model of the heart. So it'd be really nice if instead of trying to estimate those thousands of parameters, we could estimate something smaller, make for an easier inverse problem. Now, before we talk about how we did that, I'd just like to briefly talk about machine learning, because that's the technique that we applied. So if you remember, this is our rule-based model, which includes physics assumptions and rules to make a model. Then we can pass inputs to make outputs. A machine learning model is a little bit different, and I call it a data-driven model, because we start with a bunch of example inputs and outputs. We then put our data-driven model in the middle here, and we look at what outputs it's going to guess. And then we compute how far off these outputs are from the ones that we wanted it to get, right? So if we put in a bunch of pictures of cats and dogs, we could say, do you think this picture of a cat is a cat? And the machine learning or the data-driven model will say yes or no. We can say how accurate that is. And then we can use that accuracy to kind of twist all the parameters of the model until it gets more accurate. The nice thing about this is that we didn't have to define any physics along the way, and we can represent something really complex with a relatively simple model. And then we can apply it just like a rule-based model. We can give it new inputs and compute its outputs. And the specific type of machine learning model that I want to focus on, data-driven model, is going to look something like this, where we have an input and an output, and the input and the output are the same, right? And that might seem kind of useless, but let's just look under the hood real quick to see what's going on. So inside of this data-driven model, we have what's called an encoder, a latent space, and a decoder. The encoder takes the input, which is this very big thing, very complicated thing, and reduces it down to a very small thing, which is this latent space. The decoder then takes that very small thing and blows it back up again into the big thing. And then by giving this data-driven model thousands of examples of inputs and outputs, we can train it and then isolate this back half, this decoder bit. And what we're left with is an algorithm that's able to take something very small and turn it into something very big. And some of you may be able to see where I'm going with this. That's something very small might be something that's very easy to optimize over. Could we plug this into our ECGI formulation? Well, in order to do that, let's get a little more specific. Let's take a mesh of the heart, which looks like this, where we've got the gray, which is the heart tissue, and then the red, which are the blood pools. And let's generate a bunch of synthetic ischemic regions. Each of these blue blobs is a region of myocardial ischemia that we just made up. And we made thousands and thousands of these ischemic zones. And then we used them to train this encoder-decoder data-driven model. For those of you who are curious, this is the internal structure of it. I'm not going to linger on it too long, but feel free to make me come back to that slide. And then once we have it trained, we now have a way of going from a small space to this full representation of myocardial ischemia. We can now take this something smaller and make it our latent space of our autoencoder, which we can then use to generate our ischemic regions to then solve our inverse problem. And the cool benefit of this is it's not strictly just the data-driven model, right? This encoding of the ischemic regions into a latent space, that's a data-driven part, but then we also still have a rule-based physics-driven model coupled to it. So we're actually working with a combined model here. Now, of course, doing this required some technical steps. I had to go write my own finite element solver, which is something that I both do and do not recommend all of you other grad students do. Good learning experience. But how did we do, right? we went through all this work. Was it worth it? Well, what I'm going to show you here is I'm going to show you the localization error. How far away is the ischemic region that we reconstructed from how far away it should be? And we generated thousands of examples, and I split them into a training set and a testing set. The training set was used to train our model, and then the testing set was one it hadn't seen before. And first, let's look at how our baseline method did, which had 2,900 parameters, which are the locations throughout this heart model. So we can see that it had an average error of around like 10 to 15 millimeters in both the training and testing set, and pretty long tails, right? Pretty high errors in some cases. Now let's look at our data-driven model. And I get to choose how many variables I want to have, right? I get to choose the size of this latent space. So in this first case, I said, let's go real small and make it 3. And in this case, we actually do get better in a lot of cases, right? We can see that the amount of ischemic regions that were reconstructed near 0 got a lot higher. There's still some errors in there. It's not perfect, but it did pretty good. The next step was to look at even more variables. And in that case, we did even better. Like a lot of these ones were pretty close to right. There still is some long tails. When the machine learning model is wrong, it's usually very wrong. But when it's right, it's very right. And then, as a next step, obviously, you just keep pushing it. We tried 32 latent parameters, and that actually made it worse. This is interesting. There could be a lot of reasons having to do with the training of the machine learning model or the optimization in the inverse problem, and we're still working on this, and I would love to hear thoughts about it. But that's what we got so far. And what I want you to focus on in this testing set is that the baseline method did not perform as well as any of our machine learning methods at getting a lot of ischemic regions close to where they want to be. So we're, of course, we've prepared this into a submission that is forthcoming to some computational engineering journal. And with that, I just want to go over some general conclusions from my PhD work. So our overall goal was to improve ECGI. In the first aim, we developed an error correction method to correct for errors in the heart position. In the second aim, we developed and applied an uncertainty quantification technique to the ECGI inverse problem. And in the third aim, we developed a novel machine learning-based model for ECGI. We were able to reduce parameters from 2,000 down to 3 in some cases. We were able to improve the accuracy of estimating ischemic regions. And we were also able to combine machine learning and a rule-based model together. And so with that, those are the goals of my PhD. Some future studies and directions that come from this. Aim 1 from the error correction method, we would of course like to extend this to other sources of error, not just the position of the heart. We would like to refine this method to address the lingering errors that we saw. And we would of course like to apply this to some more realistic clinical data, see if it can actually be integrated into a clinical workflow. Aim 2 is pretty similar. We want to extend this to other sources of error and uncertainty. We want to apply to a specific ECGI use case or pathophysiology. We looked at uncertainty quantification generically. Now we need to pick a specific use case. And then for aim 3, we'd like to apply other machine learning architectures and approaches. And we'd like to extend this combined machine learning and traditional ECGI approach to other ECGI formulations. Overall, it seems like reasonable that we could combine all 3 of these aims into a single ECGI formulation that estimates the position of the heart, does uncertainty quantification, and leverages machine learning to localize myocardial ischemia. From this research, there's of course the 3 primary journal publications, as well as I've had the wonderful opportunity through collaborators and other work to publish a number of other studies in a number of journals. And then I've also had a large range of collaborations and work that's resulted in a number of conference papers and proceedings. And with that, I'd like to thank everyone who came here to this. Don't clap quite yet because I gotta go through some thanks and then we'll do questions. Thank all of you who came to see this, both remotely and in person. Thank all of the various people I've worked with at Scientific Computing Imaging Institute, the Cardiac Electrophysiology Group, my committee, of course, the CBRTI up on Upper Campus, Lyric in France, who let me come hang out with them over the summer, the Consortium for ECGI with many people who I've collaborated with, Medical University of Graz, Northeastern University, people in industry, and of course my funding sources. I'd like to call out specifically my committee for meeting with me very regularly and giving me lots of helpful feedback. I'd like to also call out a lot of my specific mentors who weren't just on my committee, such as Dana Brooks, who taught me a lot about math, Matthias Kleitmans, who's a leader and a role model for me in the field of ECGI. Jean-Michel Font, who I also made teach me a lot of math and has collaborated very closely with me. Wilson Good and Brian Zanger, who were very direct mentors to me when I first started this process and even now. Dr. Steinberg, who has been a recent mentor of mine, who's been helping me think about broadening my clinical application space. Marcus Foote and Blake Zimmerman, who both helped me a lot with my technical implementations in a lot of these aims. And then, of course, the people from before my time in graduate school, specifically Dr. Brian Avery, who helped encourage me to pursue research and find my passion. And Drs. Peter Conwell and Peter Nichols. Dr. Conwell was the person who told me that I wanted to go to grad school. He saw it before I did. And Dr. Nichols was my high school teacher who gave me my first inspiration and love of science. Of course, I want to thank the Cardiac Electrophysiology Group, all my fellow grad students, undergrads, and the like. I want to thank specifically a lot of people at Ski who have helped me a lot, like Dan Ski Run Man, who has helped implement all these crazy features that I asked for. Ed, who helps do all of these technical things, like makes this talk go smoothly. Deborah, Magali, Brenda, all of the Chris and Elias, and all of these people who help Ski run smoothly. I couldn't have done this without you. Special shout out to Lyric and Laura Bear and Emma, who helped me have a great time when I came out to work with them over the summer. And then, of course, all of the friends and family and friends and dogs and friends and family, and these slides are not loading very quickly. There's a lot of really high resolution pictures on these. I'll just skip to the last one, which has everyone on it. It takes a village, it takes a city, it takes a whole university, and then it also takes a pretty immense support network on top of all of that. And then, of course, the person I need to thank the most of all is my lovely and supporting wife, who's in several of these photos. Come on, you can do it. Don't use Google Slides. My lovely and supporting wife, who's built a lovely life with me and supported me throughout this entire journey. I couldn't have done it without you, and of course, without our dogs, even if they try and eat chocolate chip cookies the night before my defense. And with that, I'd love to take any questions.